సో జనరల్ సైన్స్ లో రివిజన్ లో భాగంగా మనం దిస్ టాపిక్ లో వీఆర్ గోయింగ్ టు ఫోకస్ ఆన్ జెనెటిక్స్. జెనెటిక్స్ సంబంధించి ఏరియాస్ ఇంపార్టెంట్, మనకు ఎగ్జామ్ లో ఎలాంటి క్వశ్చన్స్ వచ్చే అవకాశం ఉంది, వీఆర్ గోయింగ్ టు డిస్కస్ హియర్. సో ఫస్ట్ ఆఫ్ ఆల్ లెట్స్ బిగిన్ అవర్ డిస్కషన్ విత్ ది జెనెటిక్స్ డెఫినేషన్. డెఫినేషన్ ఏంటి, జెనెటిక్స్ అంటే ఏంటి? జెనెటిక్స్ అంటే నథింగ్ బట్ స్టడీ ఆఫ్ ఇన్హెరిటెన్స్ లేదా స్టడీ ఆఫ్ హెరిడిటరీ అంటాం. ఎస్, స్టడీ ఆఫ్ హెరిడిటరీ లేదా స్టడీ ఆఫ్ ఇన్హెరిటెన్స్ అంటే పేరెంట్స్ నుంచి మనకు కొన్ని క్యారెక్టరిస్టిక్స్ ఎలా వస్తాయి, ద ప్రాసెస్ ఇన్ విచ్ హౌ different traits or characteristics pass from parents to us. Study of such information is called inheritance and genetics, to be specific. So ee characters emai untai, the specific traits could be physical characteristics like appearance kavachu or color, height kavachu, hair color, skin color, konni mannerisms. These are all inherited from parents to children. In a similar manner, we also receive certain genetic problems from parents. Genetic disorders parents nunchi కొన్ని examples మీకు తర్వాత ఇస్తాను, can also inherit in the process of this. కనుక understanding genetics is very, very important, especially to not only gain insight into how our body function in terms of the instructions that are provided from our parents to us, at the same time to understand in how to tackle genetic diseases. Okay? Right. So let's learn some terminology and basic background to genetics. So genetics is all about, in a way, DNA. DNA stands for deoxyribonucleic acid. DNA ni manam deoxyribonucleic acid అని పిలుస్తాం. deoxyribonucleic acid అంట. Okay. So ఈ deoxyribonucleic acid made up of nucleotides. It is a chain of nucleotides. Nucleotides ante fundamental units anamata, DNA yokka units. So atla each bead like nucleotides join together to form this polymer called DNA. And on this DNA, because we normally notice that we use terms like chromosomes, genes, etc., you have to have a good understanding about how are they related to each other. So DNA is the instruction manual for our life, which we receive from our parents, right? Which is made up of, what I was telling you, is nucleotides. There are 4 important nucleotides that are adenine, thymine, cytosine, and guanine. ఇవి 4 రకాల బేసెస్ అన్నమాట. ఒక బ్రీఫ్ గా ఈ 4 పేర్లు గుర్తుపెట్టుకోండి: అడెనిన్, థైమిన్, సైటోసిన్ అండ్ గువెనిన్. ఈ బేసెస్ తో పాటు ఇంకా షుగర్, అట్లాగే ఫాస్ఫేట్, ఈ 3 కలిపి ఒక న్యూక్లియోటైడ్ తయారవుతాయి. అట్లా 4 రకాల న్యూక్లియోటైడ్స్ డిపెండింగ్ ఆన్ అడెనిన్ కానీ, థైమిన్ కానీ, సైటోసిన్ కానీ, గువెనిన్ ని బట్టి 4 రకాల న్యూక్లియోటైడ్స్ ఉంటాయి. ఓకే, దట్స్ హౌ డిఎన్ఏ ఇస్ మేడ్. బట్ దిస్ డిఎన్ఏ ఆల్దో ఇన్స్ట్రక్షన్ మాన్యువల్, మామూలుగా ఏంటంటే ఇది ఎక్కడ ఉంటుంది, న్యూక్లియస్ లోపల ఉంటుంది. డిఎన్ఏ ఇస్ ప్రెసెంట్ ఇన్సైడ్ ది సెల్స్ న్యూక్లియస్. ఏ ఫార్మాట్ లో ఉంటుంది? సో లూజ్ క్రోమాటిన్ స్ట్రక్చర్ ఫార్మాట్ లో ఉంటుంది. మామూలుగా it is found in the form of chromatin, అంటే more or less కొంచెం సెమీ ఆర్గనైజ్డ్ ఒక లూజ్ థ్రెడ్ లాగా ఉంటుంది. దాన్ని క్రోమాటిన్ అని పిలుస్తాం. దిస్ క్రోమాటిన్ డ్యూరింగ్ సెల్ డివిజన్ ప్రాసెస్, మనం ఆల్రెడీ అనుకున్నాం కొన్ని ప్రీవియస్ క్లాసెస్ లో, is this chromatin condenses, అంటే ఈ డిఎన్ఏ స్ట్రక్చర్ ఏమవుతుంది, అండర్గోస్ కండెన్సేషన్ ప్రాసెస్ టు ప్రొడ్యూస్ వాట్ ఈస్ కాల్డ్ క్రోమోజోమ్. అట్లా క్రోమోజోమ్ అనే కండెన్స్ ఫామ్ తయారవుతుంది. అంటే మనం ఇన్స్ట్రక్షన్స్ గురించి డిఎన్ఏ రూపంలో చదివినా, క్రోమోజోమ్ రూపంలో చదివినా కూడా వీఆర్ టాకింగ్ అబౌట్ ద సేమ్ థింగ్. జస్ట్ ఆర్గనైజేషన్ ఆఫ్ ఇన్ఫర్మేషన్ ఇస్ డిఫరెంట్, ఆర్గనైజేషన్ ఆఫ్ ద స్ట్రక్చర్ ఇస్ డిఫరెంట్. ఓకే, సో లైక్ దాట్ వీ హ్యావ్ క్రోమోజోమ్. ఆ క్రోమోజోమ్ మీద కానీ, డిఎన్ఏ మీద కానీ a portion of this మామూలుగా దీన్ని ఫంక్షనల్ పోర్షన్ లేదా ఫంక్షనల్ యూనిట్ ఆఫ్ డిఎన్ఏ అని పిలుస్తాం, విచ్ యాక్చువల్లీ కోడ్స్ ఇన్ఫర్మేషన్ ఫర్ ప్రోటీన్స్. ఆ రీజన్ ని, డిఎన్ఏ మీద ఉండే ఆ రీజన్ ని జీన్స్ అని పిలుస్తాం. ఓకే, సో జీన్స్ ఆర్ బేసిక్ ఫిజికల్ యూనిట్ ఆఫ్ ఇన్హెరిటెన్స్, అంటే ఫిజికల్ గా వాటి నుంచి ఇన్ఫర్మేషన్ అనేది ట్రావెల్ అవుతుంది, విచ్ ఆర్ స్మాల్ సెక్షన్స్ ఆఫ్ డిఎన్ఏ దట్ ఆర్ కోడెడ్ ఫర్ స్పెసిఫిక్ ట్రేట్స్. అంటే ఇండివిజువల్ గా కానీ, కలిసి కానీ. రియాలిటీకి వస్తే కనుక మల్టిపుల్ జీన్స్ కోడ్ చేస్తున్నాయి. కొన్ని జీన్స్ ఓన్లీ పాలిపెప్టైడ్ అంటే ఒక చైన్ ఆఫ్ ప్రోటీన్స్ నే కోడ్ చేస్తూ ఉంటుంది. అట్లా దెర్ ఇస్ లాట్ ఆఫ్ వెరైటీ. బట్ మన అండర్స్టాండింగ్, ఈ ఎగ్జామ్ పాయింట్ ఆఫ్ వ్యూలో చూస్తే కనుక సెగ్మెంట్ ఆన్ డిఎన్ఏ ఇస్ కాల్డ్ జీన్. ఓకే, సో జీన్ నుంచి ఏమొస్తుంది, మామూలుగా జీన్ అండర్గోస్ ఎక్స్ప్రెషన్ అంటాం. ఈ డిఎన్ఏ రీజన్ నుంచి మెసెంజర్ ఆర్ఎన్ఏ తయారవుతుంది న్యూక్లియస్ లో. మెసెంజర్ ఆర్ఎన్ఏ అంటే ఎప్పుడైనా ఆ పర్టికులర్ క్యారెక్టర్ ఎక్స్ప్రెస్ అవ్వాలంటే, ఫర్ ఎగ్జాంపుల్ మీరు ఇన్సులిన్ తీసుకోండి, ఇన్సులిన్ ప్రోటీన్ ప్రొడ్యూస్ అవ్వాలంటే మనకు, సో డిఎన్ఏలో ఆ పర్టికులర్ రీజన్ యాక్టివేట్ అవ్వాలి. అంటే అక్కడ ఉన్న రీజన్ ట్రాన్స్క్రైబ్ అయ్యి, అంటే ఆ ఇన్ఫర్మేషన్, డిఎన్ఏ ఫామ్ లో ఉన్న ఇన్ఫర్మేషన్ ఆర్ఎన్ఏ ఫామ్ లోకి కన్వర్ట్ చేయాలి. అప్పుడు దాన్ని మనం ట్రాన్స్క్రిప్షన్ అని పిలుస్తాం. ఆ ట్రాన్స్క్రిప్షన్ ప్రొడక్ట్ ఏదైతే ఎంఆర్ఎన్ఏ ఉందో, దట్ ఎంఆర్ఎన్ఏ గోస్ ఇన్ టు ది సైటోప్లాజం లుకింగ్ ఫర్ రైబోసోమ్స్. And ribosomes, which are protein synthesizing factories, would identify the messenger RNA and start reading it. When ribosomes read messenger RNA, they start bringing amino acids. Amino acids are the building blocks of proteins. ఎట్లా న్యూక్లియోటైడ్స్ ఆర్ బిల్డింగ్ బ్లాక్స్ ఆఫ్ డిఎన్ఏను, అలాగే అమైనో యాసిడ్స్ ఆర్ బిల్డింగ్ బ్లాక్స్ ఆఫ్ ప్రోటీన్స్. జీన్స్ అనేవి ఇక్కడ కోడ్స్ అయితే, ప్రోటీన్స్ అనేవి ప్రొడక్ట్స్. అట్లా ఈ డిఎన్ఏ రీజన్ విచ్ ఇస్ కాల్డ్ జీన్, ఫ్రమ్ దిస్ త్రూ ట్రాన్స్క్రిప్షన్ మెసెంజర్ ఆర్ఎన్ఏ ఇస్ మేడ్. ఆ మెసెంజర్ ఆర్ఎన్ఏ ఇస్ రెడ్ బై రైబోసోమ్ ఇన్ సైటోప్లాజం. ఇలా మీరు లొకేషన్స్ అడగొచ్చు. ఇదంతా కూడా ప్రొడక్షన్ ఆఫ్ ప్రెసెన్స్ ఆఫ్ డిఎన్ఏ, ప్రొడక్షన్ ఆఫ్ ఆర్ఎన్ఏ హ్యాపెన్స్ ఇన్సైడ్ ద న్యూక్లియస్. బట్ ఈ మెసేజ్ బయటకు రావాలి, రైబోసోమ్స్ ఆర్ అవుట్ సైడ్ ద న్యూక్లియస్. దట్ ఈస్ వేర్ ఇన్ఫర్మేషన్ ప్రెసెంట్ ఇన్ ద ఫార్మ్ ఆఫ్ మెసెంజర్ ఆర్ఎన్ఏ ఈస్ ట్రాన్స్లేటెడ్. ఈ ప్రాసెస్ ని ట్రాన్స్లేషన్ అంటాం, ఇంటూ ప్రోటీన్స్. అట్లా ప్రోటీన్స్ అనేవి తయారవుతాయి. అట్లా ప్రోటీన్స్ వుడ్ గివ్ రైస్ టు డిఫరెంట్ క్యారెక్టరిస్టిక్స్. అయితే ఫంక్షనల్ ఆస్పెక్ట్స్ ఉండొచ్చు, స్ట్రక్చరల్ ఆస్పెక్ట్స్ ఉండొచ్చు. స్ట్రక్చరల్ అంటే మన మజిల్స్ తయారవడం, బాడీ పార్ట్స్ తయారవడం. ఫంక్షనల్ అనుకోండి, ఇన్సులిన్ ఎగ్జాంపుల్ చెప్పాను కదా, ఇన్సులిన్ బ్లడ్ గ్లూకోజ్ లెవెల్ ని తగ్గించడం. అట్లా ప్రొడక్ట్స్ మనం తయారు చేసుకుంటూ ఉంటాం. రైట్. సో ఇది ఇలా బేసిక్ గా మన ఇన్స్ట్రక్షన్స్ ఆర్ స్టోర్డ్ ఇన్ ద ఫార్మ్ ఆఫ్ డిఎన్ఏ. ఈ క్రోమోజోమ్, జీన్ అనే పదాలకి లింక్ అర్థం చేసుకోండి. రైట్. సో డిఎన్ఏ స్ట్రక్చర్ ఎవరు కనుక్కున్నారంటే క్వశ్చన్ అడగవచ్చు. డిఎన్ఏ స్ట్రక్చర్ వాజ్ గివెన్ బై ఫ్రాన్సిస్ క్రిక్ అనే శాస్త్రవేత్త, అట్లాగే జేమ్స్ వాట్సన్ అనే శాస్త్రవేత్త, మౌరిస్ విల్కిన్స్. ఇంకా కొందరి కంట్రిబ్యూషన్ కూడా దీనికి ఉంది. రోజలిన్ ఫ్రాంక్లిన్ అంటారు, రోజలిన్ ఫ్రాంక్లిన్. షీ ఆల్సో కంట్రిబ్యూటెడ్ టు దిస్, బట్ షీ డిడ్ నాట్ రిసీవ్ నోబెల్ ప్రైజ్ ఫర్ దిస్. వీళ్ళకి 1962లో నోబెల్ ప్రైజ్ ఇన్ ఫిజియాలజీ అండ్ మెడిసిన్ కి ఈ డిఎన్ఏ స్ట్రక్చర్ కనుక్కున్నందుకు గాను ఇవ్వడం జరిగింది. సో ఈ ఇన్ఫర్మేషన్ గుర్తుపెట్టుకోండి. హూ రిసీవ్ నోబెల్ ప్రైజ్ ఆర్ హూ గేవ్ డిఎన్ఏ స్ట్రక్చర్ ఇన్ స్ట్రక్చరల్ ఇన్ఫర్మేషన్. ఓకే, ఇది బ్రీఫ్ బ్యాక్గ్రౌండ్ మనకి. సో ఎన్ని క్రోమోజోమ్స్ ఉంటాయి హ్యూమన్స్ లో? హ్యూమన్స్ హ్యావ్ 2 sets of 23 క్రోమోజోమ్స్. 23 క్రోమోజోములు ఉంటాయి. అంటే ప్రతి పేరెంట్ నుంచి కూడా మనకు 23 క్రోమోజోమ్స్ వస్తాయి. అంటే ఆల్ టుగెదర్ ఒక మదర్ నుంచి ఫాదర్ నుంచి కలిపి మనకు 46 క్రోమోజోమ్స్. 2 times 23 క్రోమోజోమ్స్, 46 క్రోమోజోమ్స్ వస్తాయి. అట్లా 2 కాపీస్ ఆఫ్ ఈచ్ క్రోమోజోమ్ ఉంటుంది కనుక మనం డిప్లాయిడ్ సెల్స్, డిప్లాయిడ్ అంటే 2 sets of ఈచ్ క్రోమోజోమ్ ఉంటాయి కనుక డిప్లాయిడ్. మీకు హ్యాప్లాయిడ్ అంటే ఏంటి, డిప్లాయిడ్ అంటే ఏంటి అనేది వి హావ్ టాక్డ్ అబౌట్ ఇన్ ది సెల్ డివిజన్ ప్రాసెస్ లో మీకు చెప్పాను. ఓకే, సో ఇది డిప్లాయిడ్ సెల్ నంబర్. క్వశ్చన్స్ అట్లా ఫ్రేమ్ చేస్తే వాట్ ఈస్ ద డిప్లాయిడ్ నెంబర్ ఆఫ్ క్రోమోజోమ్స్ ఇన్ హ్యూమన్స్ అంటే 46 రాయాలి. ఇన్ కేస్ ఇఫ్ ద క్వశ్చన్ ఈస్ ఫ్రేమ్ లైక్ దిస్, వాట్ ఈస్ ది హ్యాప్లాయిడ్ నెంబర్ ఆఫ్ క్రోమోజోమ్స్ అంటే ఇట్ ఈస్ 23 not 46. అది గుర్తుపెట్టు. ఓకే, రైట్. సో అండ్ ఈ క్రోమోజోమ్స్ అయితే 23 pairs ఆఫ్ క్రోమోజోమ్స్ ఉన్నాయో, అందులో 22 pairs ఆఫ్ క్రోమోజోమ్స్ ని ఆటోజోమ్స్ అని పిలుస్తాం. 22 pairs of chromosomes are called autosomes అన్నమాట. 22 pairs అంటే 44 ఆటోజోమ్స్ ఉంటాయి. ఇవేం చేస్తాయి? దీస్ యాక్చువల్లీ కంట్రోల్ సోమాటిక్ క్యారెక్టరిస్టిక్స్, అంటే బాడీలీ క్యారెక్టరిస్టిక్స్, బాడీలో వెరైటీ ఆఫ్ పార్ట్స్ కావాలన్నా, గ్రోత్ లాంటి సంబంధించిన విషయాలన్నా, అంటే సెక్స్ కి సంబంధించి, రీప్రొడక్షన్ సంబంధించి, జెండర్ డిటర్మినేషన్ సంబంధించిన వాటిని పక్కన పెడితే మిగతావి ఆటోజోమ్స్ వుడ్ టేక్ కేర్ ఆఫ్. అందులో క్లియర్ గా మనం డిమార్కేట్ చేయడం. వాట్ వీ హావ్ టు రిమెంబర్, టెక్స్ట్ బుక్ నాలెడ్జ్ ప్రకారం ఆటోజోమ్స్ అంటే ఇవి, అల్లోజోమ్స్ అంటే ఇవి.
సో ఇంకా కొన్ని ఫంక్షన్స్ లైక్ గ్రోత్ సంబంధించిన ఇలాంటివి కూడా అల్లోజోమ్స్ రోల్ ప్లే చేసే అవకాశం ఉంది. బట్ మొరల్లెస్ గా మనం ఈ ఎగ్జామ్ పాయింట్ ఆఫ్ వ్యూలో గుర్తుపెట్టుకోవాల్సింది ఏంటంటే, అల్లోజోమ్స్ అనేవి లైక్ మోస్ట్లీ రిప్రొడక్టివ్ రిలేటెడ్ సంబంధించిన క్యారెక్టర్స్ కంట్రోల్ చేస్తాయి, వేరేజ్ ఆటోజోమ్స్ అనేవి దే కంట్రోల్ అదర్ బాడీలీ క్యారెక్టరిస్టిక్స్ అండ్ బాడీలీ రిలేటెడ్ కాంపోనెంట్స్ అన్నమాట. రైట్, ఓవరాల్ గా ఆటోజోమ్స్, అల్లోజోమ్స్ గుర్తుపెట్టుకోండి. నెక్స్ట్ విల్ టాక్ అబౌట్ ప్రిన్సిపల్స్ ఆఫ్ ఇన్హెరిటెన్స్. సో జెనెటిక్స్ లో కీగా మనం చదువుకునేది ఈ దోస్ లాస్ దట్ ఆర్ గివెన్ బై హూ ఇస్ కన్సిడర్డ్ టు బి ఫాదర్ ఆఫ్ జెనెటిక్స్. ఫాదర్ ఆఫ్ జెనెటిక్స్ ఎవరు? ఈజ్ గ్రిగర్ మెండల్. గ్రిగర్ మెండల్ ఈజ్ అన్ ఆస్ట్రియన్ మాంక్. ఆయన గార్డెన్ పీ ప్లాంట్ మీద వర్క్ చేశారు. ఈ వర్క్ ఆన్ గార్డెన్ పీ ప్లాంట్. సో సైంటిఫిక్ నేమ్ ఆఫ్ గార్డెన్ పీ ప్లాంట్ ఏంటి? ఇట్ ఇస్ పైసమ్ సెటైవం. Paisam sativa. ఈ గార్డెన్ పీ ప్లాంట్ మీద వర్క్ చేసి he gave principles of inheritance. అంటే ఈ characters what type of rules or laws do they follow. That is what he figured out using this garden pea plant. I don't think the exam lo meeku specific ga adagaru, but in case if you have to remember 7 characteristics that he has worked on. ఈ పైసం సెటావిక్ సంబంధించిన సీడ్స్ కలర్ కానీ, సీడ్ షేప్, సీడ్ కోట్ కలర్, length of the stem, pod color, pod shape, flower position. These are the type of things he had worked on to establish the laws of inheritance. Right. So first we'll begin discussing the terminology related to that. So in exams we have been seeing that they can ask questions on like what does this term mean, what is it referred to, what is the right statement that describes it. So locus, if they are asking about locus, locus is the location of a gene on DNA. So in a way this can be considered as physical address of a gene. So where exactly a gene is present on the DNA, such a region is called locus. Then we already talked about gene. Gene is a functional unit or functional segment of DNA. In with respect to this gene, in genetics we also hear a term called alleles. So what are alleles? Alleles are considered to be alternative forms of a gene. No, that actually are controlling alternative forms of a character. Meaning, so there is height. Let's say plants, some plants are tall, some plants are dwarf. Same genes may control this character. There may be a version of gene that controls tallness of the plant, a version of the gene or combination of which may result in dwarf plants. So like that, they probably found in the same location, same locus on different chromosomes. Mother యొక్క chromosome 2 లో ఉందనుకుందాం ఈ locus లో, ఫాదర్ chromosome 2 లో ఆల్టర్నేట్ ఫామ్ ఉండే అవకాశం ఉంటుంది. ఇట్లా ఒక క్యారెక్టర్ యొక్క ఆల్టర్నేట్ ఫార్మ్స్ ని కంట్రోల్ చేసే కాంపోనెంట్స్ ని అలీల్స్ అంటాం. బేసికల్ గా అలీల్స్ ఆర్ ప్రెసెంట్ ఆన్ 2 హోమోలాగస్ క్రోమోజోమ్స్. ఫాదర్ క్రోమోజోమ్ మీద ఒకటి, మదర్ క్రోమోజోమ్ మీద ఒకటి ఉండే అవకాశం ఉంటుంది. లేదా దే కెన్ బి సేమ్ డిపెండింగ్ ఆన్ what type of gene that given parent is carrying. Those are alleles. Okay. Next, we hear about what is called homozygous condition, heterozygous condition. Homozygous condition is where two alleles, same alleles are present on corresponding location, corresponding loci. Like Maulga, as per Mendel, if an allele responsible for tallness of a plant, it is considered to be capital T. He designated it as capital T. So like that, mother plant and parent plant, like paternal plant, both may carry same capital T's. If they happen to have same alleles in corresponding locations, these are called, this is called homozygous condition. And whereas if two different alleles, okay, meaning two alleles like capital T, small t is being present, small t representing here the dwarfness, then one capital T, one small t. So this is heterozygous condition, when two different alleles are present in the corresponding loci, like capital T and small t. Whereas if two small t's are present, it is similar to this condition. So this is also homozygous condition for a different allele. So dominant, we'll talk about this in a couple of minutes. So dominant allele and recessive allele, we refer to them as. So this is homozygous condition for dominant allele. This is recessive allele for that condition. So what is dominant allele or recessive allele? Dominant allele is an allele that expresses here, in this case, capital T, small t. If we denote, capital T codes for tallness or controls tallness, whereas small t controls dwarfness. In this condition, which one should express? Because there are both capital T, small t, the one that is responsible for tallness of a plant, the one responsible for dwarfness of a plant. Then which one should actually express? As per Mendel's observation, the characteristic of capital T gets expressed despite of the presence of small t. Then he called it dominance. He called it, this is a dominant allele, and the one that is undergoing, that is not being able to express itself in presence of capital T, is called recessive allele. So these are recessive alleles, these are dominant alleles. In a heterozygous condition, only dominant allele expresses if they obey this law of Mendel, which can be seen clearly with respect to blood groups. Blood groups, in blood group A, AA combination is homozygous. Okay, BB, here these are called little different from normal dominant alleles. These are called codominant alleles that we are going to talk about next. Okay, heterozygous condition, you can see both A and B alleles may be present. Here, they are not dominating each other. They both express, which is a different case of, inheritance. Right. Next, what is genotype and phenotype? This has been asked in group 1 exam once. So genotype refers to what? So genotype refers to the internal genetic combination for a given character. So it could be said as genetic makeup of an organism. Like if you see, there are tall plants. These tall plants may be because of two capital T's being present. The capital T is a genotype of it. Whereas the same tallness can also be seen because of heterozygous condition. Here we are referring to genotype of that capital T, small t is genotype. Whereas dwarfness that we notice in coming crosses, that small t, because it's a recessive allele, only when two recessive alleles are together, present together, only then dwarfness is expressed. Okay, so this combination of letters, alleles, is called genotype. Whereas phenotype is what we see outside, or it is individual's appearance. What we see externally is called phenotype. So this tallness, dwarfness are referred to phenotypes. So know the difference between genotype and phenotype. Genotype is the internal genetic makeup and phenotype is the external appearance of a character. Okay, I already talked to you about dominant alleles and recessive alleles. One thing I would like to add here, if it is a disease condition, because these characteristics could be even diseases, mutations passing on from parents to children, resulting in a disease condition or genetic condition. So in those cases, if there are two same copies are present which are defective, okay, so then certain diseases which are considered to be recessive alleles express. Whereas if it is case of dominant alleles, doesn't matter. Even one defective allele is present, other one is normal allele, because we normally get two alleles, one from each parent. So if it is dominant form of inheritance or condition, so irrespective of having one normal allele, one defective allele can still cause problem. Okay, so like that, dominant allele, recessive alleles can also be seen in terms of diseases. Although dominant allele, genetic diseases that inherit through dominant fashion, in dominant fashion, are rare compared to recessive allele, or those that are progressed through or inherited through recessive alleles. So these are the terms. Next, I'm going to give you briefly the inheritance principles given by Mendel. The first one we kind of already talked about, which is called, first law is called law of dominance. So what happens here? So one dominant allele, if there is a dominant allele that overpowers or that dominates the recessive allele. In this particular very famous case or example worked out by Richard Mendel. So if you have tall plants, homozygous tall plants are called, two capital T's will be present, and homozygous dwarf plants, two small t's will be present. If you cross those plants, which are homozygous for both of these conditions. So normally we write gametes. So what type of gametes are possible? Both are capital T's, only one type of gamete is possible, which is the one with capital T. Here, only small t type of gametes are possible. So small t. If you combine them, this is a female and this is a male version or vice versa. If you combine them, the only product will be capital T, small t, where genotype will be capital T, small t. Phenotype will be tall, all tall. All how many number of plants are produced, they will be tall in nature. Okay, so like that, we notice that here in this case, when you take pure lines or homozygous lines, if you cross them, if it is a dominant character, all F1 plants, F1 means first generation plants, or we can also refer it to as filial generation. All of them will be tall and heterozygous in nature. That is what Mendel has figured out. And when you are only observing single character like this, your height, then it is called monohybrid cross. So law of dominance, Mendel proved through this process that irrespective of presence of one small t here, the capital T is the one that got to express here. That is what is called dominance, because capital T is dominating over small t. Only that is why, despite of using two opposite parents, only tall character has been expressed. Okay, this is how Mendel came up with the first law of dominance. Okay, and Mendel of course did perform few tests, because he didn't know, we cannot see it outside whether it is a dominant or recessive or homozygous condition or heterozygous condition. So Mendel has performed what is called test cross for this. Okay, I'll come back to this idea of test cross, but for now, just remember that we could do a cross between two, F1 generation plants. That is what he had done. I'll tell you why this has been done. But next, I'll briefly talk to you about incomplete dominance. Dominance, that is, unlike what we have discussed in heterozygous condition, none of these genes or alleles dominate other. So what happens here in this particular case, when one allele of a gene is not completely dominant over the other, then what happens in the F1 category? Like if you take red color, plants with red color flowers, plants with white color flowers. Red is supposed to be dominant, or we consider it to be the dominant version. So it is written using capital R, and white is small r. But interestingly, in the hybrids, or when an F1 generation has formed, and where it's supposed to be one capital R coming from here, one small r coming from here, it is intermediate. It is expressing intermediate characteristic between these two colors. Between red and white, it is actually showed pink color. So neither of them are dominating. It's an intermediate character between these two. In such cases, it is called incomplete dominance. This is seen in a plant called snapdragons. Snapdragons actually tend to show these characters. Similarly, you can even see that in incomplete dominance, flowers, colors of animals can be in between both the parents. In such case, we are looking into incomplete dominance. Okay, pink flowers are produced when you cross red ones, plants with red ones, plants with white ones. So if you see the ratios, because here we've talked about in the F1 generation, 100% all the plants are so dominant, express dominant character. But if at all you cross in the next generation heterozygotes, capital T small t with capital T small t, what you would notice if you write the gametes, this normally we see gametes separate from each other during meiosis process. This capital T will go into one gamete, small t will go into another gamete. Same happens here on the other side. This is with other, like if it is male, here we talk about female plant then. So like opposite genders, you notice same gametes gets formed. When you write the combinations of this, like capital T with capital T, okay, and capital T with small t, or one capital T with small t, or small t with small t, what you will notice is this is the combination of things that you would normally find. Where the capital T, of course, is tall. Wherever at least one capital T is present, these all three are tall plants, and this is dwarf plant. If you write ratio here, like what you phenotypically see, there are 3 tall plants and 1 dwarf plant. So this ratio in second generation of Mendel's monohybrid crosses would result in 3:1 ratio. For every 3 tall plants, 1 dwarf plant will be there. If at all you are using hybrids taken from the first generation. Same thing if they ask you about what is a monohybrid cross genotypic ratio given by Mendel. So 1 homozygous, 2 heterozygous dominant, 1 homozygous recessive. So 1 of this, so this is 1, and 2 of capital T small t's, and 1 of small t small t. So genotypic ratio is 1:2:1 in case of monohybrid cross. Let's say in incomplete dominance, how do you know this is incomplete dominance? One is visually, phenotypically you see it has intermediate color. And ratio wise, if you notice, phenotypic genotypic ratios will be same, because in this particular case we have seen that the phenotypic ratio is 3, because although these are heterozygotes, they all express to be tall. But here heterozygotes would be expressed as pink. So what you would notice in F2 generation here is 1:2:1 ratio. Phenotypes also 1:2:1. So 1 red, 1 white, and 2 pink. So that's how ratio would differ in case of codominance, incomplete dominance. There is another exception to Mendel's dominance concept. This is called codominance. In incomplete, we have seen both actually merge to give rise to intermediate characteristics. Here, they both express co-dominate. They both dominate each other. 2 alleles of a gene equally expressive and equally dominant. Very well known example is human blood groups. Let's say mother has A blood group, father has B blood group. There's a possibility that children can be A, B blood groups. So A, B both express. None of them are dominating each other. That is a very good example of codominance. So if you're asked which of the following is an example of codominance, then AB is one very good example. Blood groups will be very good example. Similarly, you can also see this happening in flowers. In this particular case of flower, red flower, plants with red flowers are crossed with plants with white flowers. And what you notice is not pink color as a case with incomplete dominance, but few of them are white, few of them are red. Both are expressing together or simultaneously. This can also be seen on the coat color or skin color of animals. like we see cows, they have black and white patches or black color skin, white patches, vice versa. Could be seen also example of codominance. So both incomplete dominance and codominance are the exceptions to Mendel's law of dominance. The second law given by Mendel is law of segregation. Law of segregation. So what is law of segregation? What does it talk about? Segregation means separation. Segregation means separation. Okay, so we kind of talked about this. So when gametes are formed, alleles from two parents will separate, like capital T. That is a parent generation that we have seen here. Parent generation, it is, let's say, capital T small t, like capital T small t. So what happens? One capital T goes into one gamete, another allele small t goes into another gamete. This is what is called segregation. So alleles separate from each other during gamete formation. That is what is given by Mendel in his second law. A diploid individual possesses a pair of alleles, either capital T, capital T small t, or small t, for any particular trait, which separate from each other during gamete formation. This is what is called law of segregation. So each gamete only receives one allele from the original gene. So based on this, Mendel found out, because when dominant characters are studied, children, like basically F1 generation, we have not seen any dwarf plants. But because of segregation, the small t got hidden, and because of segregation, the small t could pair with another small t, giving rise to dwarf plants. So this let Mendel figured out that segregation is taking place. Small t is not going anywhere, it is hidden. In absence of the capital T, along with another small t, this gets expressed. So recessive character gets expressed when it comes back. And this is possible because of segregation. Okay, that is what Mendel has explained in his second law. Third law is called law of independent assortment. So segregation normally happens in irrespective of this. There can be some physical problems where chromosomes are attached. Otherwise, segregation can happen unanimously. Next, third law of independent assortment, it is called. So far, we have seen monohybrid crosses. But what if you perform dihybrid crosses? Meaning, if you observe two different characteristics at the same time. Do they influence each other? Okay, like in this particular case, Mendel performed dihybrid crosses. Observe two traits. Like in this particular case, you notice that we're talking about color of the seed as well as shape of the seed. So color of the seed, either seeds can be yellow or green. Shape of the seeds, they could be round or wrinkled shape. So here it's supposed to read two capital Y's and two capital R's. There is a typo here. All supposed to be capitals. So capital R is round, capital Y is yellow. Yellow round are dominant characters over yellow and green. Green and wrinkled. So when you perform such a cross, you don't have to remember these details here. But what you notice is Mendel actually got a phenotype of 9:3:3:1. Mendel got. Okay, so how many we have here? Basically, we have 4 into 4, 16 that we have seen. What type of phenotypic ratio we got for monohybrid crosses? Monohybrid crosses is 1:2:1. Out of every 4 plants. Okay, so 1 dominant, 1 recessive, and 2 intermediate. Right, 2 intermediate we normally find. So for phenotype, it's usually 3:1. For genotype, 1:2:1 is what we have seen. So 3:1 you find. Here, the total is 16. Right? So if you can compare phenotype with phenotype, 3:1 ratio with the 9:3:3:1. What do you notice? The numbers may look different, but indeed they are same. Okay? Because we are considering different combinations here, it appears to show this, varying numbers, but actually they mean the same. How do you know that? So 9:3, basically if you see yellow. So one particular character if you see, so what you notice here, out of 16, like let's say these characteristics, 3 and 1 are same. So 3 plus 1 is 4. 4 out of 16 show this one green color character. Same thing here, 3 is to 1, both wrinkled ones. So 25% of them, 25%, 4 by 16 is 25%, right? So 25% of them are recessive, and 12 out of 16, 9 plus 3, 12 out of 16 are dominant. So 75% dominant, that is 3 is to 1. So ratio has not changed, meaning they behave as if, they are independent of each other. So one character has no influence on the other character. That is why through Dihybrid process, Mendel has shown the law called law of independent assortment. Separate genes for separate characteristics are passed independently of one another from parents to children. So phenotypic ratios have not really changed. This is what Mendel has figured out, indicating that two characters controlled by two different genes tend to not influence each other. But there are exceptions to that. Sometimes what happens, if those genes happen to be very close to each other, they tend to inherit together. Such a concept is called linkage. So exception to this can happen. It is called linked genes or linkage, it is known as. Okay, right. So remember phenotypic ratios, genotypic ratios will be much higher for this, which I don't think would be asked in the exam. But maybe remotely there is a possibility of asking phenotypic ratio comparison between monohybrid cross and dihybrid cross. So like that, Mendel has given these three common laws. The other term that I want you to remember about is the difference between back cross and test cross. So in the first case, we have noticed that we do not know whether it is homozygous condition or heterozygous condition, because two alleles got expressed here. Right? In this particular case, monohybrid cross, so it is capital T, small t. Tallness could be because of two capital T's or one capital T, one small t. To find out what is the genotype of this, Mendel had performed what is called test cross, which is a type of back cross. So what is the key difference between back cross and test cross? So back cross is basically using the F1 plant, that is the progeny, crossed with either of the parents. You are crossing it back with one of the parents. Then it is called back cross. Test cross is if you are actually crossing it with the homologous or homozygous recessive genotype. It was to read as homozygous recessive genotype. Meaning, if you actually take capital T, small t, that is F1, crossed with the, like as you see here, capital T, small t, crossed with the homozygous recessive parent, then it is called test cross. It is called test cross because it can reveal the genotype. It can tell you whether, let's say, as you see here, it is capital T, small t, it is heterozygous. Then what you will notice is 50% of the recessive characteristics will show. Whereas if it is a dominant character, so you only notice 25% of the children having this character. Okay. If it is two capital T's, that would be the case with dominant parent, capital T. Then what happens? All of them will be, tall, because one parent is giving two capital T, so there is no chance of small t getting expressed. Okay, so what are we talking about here? Test cross would differentiate whether it is the dominant allele, whether it is homozygous or heterozygous condition. Back cross, if you are considering another parent, may not reveal that information. Like that, Mendel talks about back crosses and test crosses to figure out the genotypes of the plants that got produced in F1 generation. So the three laws that we have talked about here given by Mendel that you need to be familiar about. Briefly know those laws and statements related to them. Right, what is the use of Mendelian information? So one, although not true with all characteristics that we carry in us, but Mendel laws do hold true for a majority of our characteristics. There are many examples that you normally see in humans which also follow Mendel laws. Like some people have attached earlobes. Here, earlobe is attached. Some people actually have, widow's peak. Basically, here there is a kind of an extension that you notice, widow's peak it is called. So there are several characteristics that follows Mendelian laws. But not all of them, because we have certain characteristics which are complex. They are controlled by not one but multiple genes. Their things may vary. Okay, so I don't think we need to get into much more details than this here for prelim's sake. So what else probably are important here? This information can also be used in following genetic diseases. what pattern are they following. So there are someone called genetic counselors who who understand this phenomena and counsel people depending on the nature of the genetic disease, nature of the inheritance pattern. So they may guide people whether to go with children or not if they are having certain deleterious alleles or defective alleles which can cause problems. They can make a choice, they can make the decision whether they should go with it or not. To guide these people, they follow the same inheritance pattern like we've so far studied, like what will be the F1 generation, what percentage of children would get the disease. Such things are possible. I'll give you an example of that in a couple of minutes, but for now, remember that is one of the benefit or significance of studying Mendelian characteristics. Okay, so next we'll talk about, I think one possibility is asking number of chromosomes being present in organisms. Some of the examples of number of chromosomes are given here. Like viruses, they don't have any concept of chromosomes. They normally have few genes, stretch of genes. Bacteria contains 1 chromosome. And Drosophila, fruit fly, contains 8 chromosomes. Rice, which is Oryza sativa, contains 24. Tomato contains 24. Okay, and hence they actually contain, in this case, 78. Mouse contains 40. And some primates, some contain 78, some contain 48. Okay, in this particular case, we are talking about dogs, of course not primates, and dogs have 78 chromosomes. So chimpanzees carry 48 chromosomes. Humans carry 46 chromosomes. Like that, there is a varied number of chromosomes, and all these numbers that I've given here is a diploid number of chromosomes. Okay, so there's a range, and there is no correlation between complexity. The dogs are less complex than us. They carry more number of chromosomes. Same thing with the primates, like chimpanzees. So some numbers are probably important. Otherwise, remember, these number of chromosomes are not fixed. Right. Next, we'll talk about genetic abnormalities. What type of abnormalities are present? A brief idea about them. So problems can happen at the chromosome level. Problems can happen at the chromosome level. Meaning, if you have organisms, okay, their chromosomes that are present in them may vary in terms of structures. Some chromosomes may lose parts of them. These are called deletions. Some chromosome may have excess portion of a chromosome, excess portion of certain regions. This is called duplication. Some may get inverted. So chromosome order may change. It is called inversion. Okay. And another one is translocation. Translocation is where chromosome part of one or part of the segment of one chromosome may get exchanged with another chromosome. In this particular case, this green chromosome, chromosome 20, has received a portion of chromosome 4. So they are translocating. These pieces are translocated. They move from one region to the next region. These are the common chromosomal structural abnormalities, structural change. Similarly, we do have numerical problems. I think one disease, if you have to remember, it is called Cry-Du-Chart syndrome. Cry-Du-Chart syndrome. Cry-Du-Chart syndrome. Okay, so because of deletion of chromosome, one of the chromosomes, chromosome number 5, when it gets deleted, it results in the disease called Cry-Du-Chart syndrome. Okay, next we'll talk about, cancers can also happen. Cancers can also happen, usually translocation. Exchange of pieces are being seen in certain type of cancers. This is about structural changes. Numerical changes can also happen. Sometimes instead of having 46 chromosomes, we may have a chromosome less or a chromosome more. That is possible. But we never show polyploidy in humans. Plants, they may carry polyploidy, meaning poly means many, ploidy, sets of chromosomes they may carry. Like they may carry 3 times the number of chromosomes, 4 times the number of chromosomes. Such things are possible in plants. But humans, we can't tolerate them. So what we can tolerate is maybe 1 chromosome less or maybe 1 chromosome more. That is what we notice here. What is a condition where instead of 46 chromosomes, we carry 45? Especially here, what happens? Females, instead of carrying 2X chromosomes, they carry only 1X chromosome. 1X chromosome is missing in them. This condition is called Turner syndrome. Turner syndrome. Sometimes we may have more than 46, so 47 chromosomes. Here, in this particular case, instead of X and Y, there is extra X chromosome. This is called Klinefelter syndrome. Okay, like that. Similarly, we have 21 trisomy, meaning one extra 21 chromosomes may be present. This is called Down syndrome. Downs. What happens here? 3 times 21 chromosomes are present. This is called 21 trisomy. So these are examples. If they ask which of the following are genetic problems or chromosomal abnormalities, know the name. I don't think they'll get into the details here. Klinefelters, Turner's, Down syndrome, etc. are names of chromosomal abnormalities where number of chromosomes are different, but usually either one more or one less. Another common problem that we see related to genetics is, or genetic related problems is, those that happen because of mutations. So by definition, what is mutation? Mutation can be seen in a very simple sense as change in DNA sequence. Usually textbooks consider it as heritable change, sudden heritable change. But more or less any change in DNA is considered to be mutation. Some are heritable, some are not. So what is change here? So notice here, in this particular sequence, this is nucleotide sequence AGG, CTT, TGG. So this T that is highlighted here has changed in this strand. So instead of T, it became G. The meaning of that codon changes. Protein that is being produced from it is affected. Okay, like that, this is called a mutation. A change in DNA sequence. What causes this mutation? During DNA replication, when DNA is getting copied, these errors may be introduced, or some mutagens are present which induce mutation, like ionizing radiation. UV rays are known as ionizing radiations. They can cause mutations. Some chemicals like pesticides can introduce mutations. Viruses, some viruses like hepatitis virus, they can introduce mutations and lead to liver cancer. So these are examples of those that introduce mutations. These are called mutagens, mutation causing agents. Okay, such mutations may lead to genetic problems. So I'll explain them with examples here. So genetic diseases, depending on where the mutation is, where the change is, either they can be called as autosomal disorders or sex chromosomal disorders or sex chromosomal diseases. So out of one of the 22 chromosomes may have an effect, may carry a mutation in autosomes. Well-known example is sickle cell anemia. Sickle cell anemia. Sickle cell anemia is because of a genetic defect or mutation happening on one of the autosomes. And the way it expresses or inherits is recessive, autosomal recessive. Meaning, I told you, recessive characters require two same defective copies, homozygous condition. So two defective copies would result in the disease. Okay. So in sickle cell anemia, what is affected? Sickle cell anemia, RBC shape is affected. Our normal RBC shape is disc shape. What happens because of this sickle cell mutation? The shape of the RBC goes from disc to sickled shape. It's like crescent shape they form. And when they form crescent shape, they can't easily flow in the blood vessels. They can't easily squeeze through the tiny blood vessels. They may stuck. They may block the flow. Like that, they may have negative impact on the people who carry it, especially those who carry two defective copies. Like one received from each parent cannot survive after certain age. Okay, so this shape of the RBC is affected and oxygen transport hence is affected in sickle cell anemia case. Another one is well-known one is thalassemia. Thalassemia, very common disorder in India. It's also genetic disorder. What happens here? There are defects. If you see normal RBC should look like this. In thalassemia, there are defective RBC because there is a mutation in hemoglobin, results in defective RBC. And because RBC is defected, our body removes those defective RBC, resulting in anemia. So it results in anemic condition. Thalassemia results in anemia or anemic condition, which is also autosomal recessive disorder. So meaning two defective alleles are needed for resulting in this condition. Right. Similarly, we find mutations on sex chromosomes, like we have either X or Y chromosomes, right? So those that actually distributed or transmitted because of mutations on X are called X-linked diseases. Those that get transmitted through Y-linked chromosomes, Y and genes present on Y chromosomes are called Y-linked. There are not many examples of Y chromosomes. So what I want you to remember is X-linked diseases. Very well known example is hemophilia. What happens in hemophilia? So So there is a defect in one of the blood clotting enzymes, because blood clotting requires several proteins and enzymes, and one of them may carry mutation. It won't be functional, so no clot gets formed. So people who are suffering from hemophilia, there is no clotting that takes place. So remember, hemophilia is also genetic disease related to X chromosome. Similarly, there is something called color blindness, also linked to X chromosome. The defective allele is present on X chromosome. Most common form of color blindness is red-green color blindness, where individual with this genetic mutation cannot differentiate red from green, and basically they have gray color instead of these colors. So these are examples for X-linked disorders. Okay, I'm going to briefly talk to you about a problem, because I have seen in one of the competitive exams they asked a problem to solve, like gave a genetic condition and asked how many children, what percentage of children are affected by this. Look at this. So let's say a colorblind woman got married to a normal man. Colorblind women got married to a normal man. So mother is colorblind, and colorblindness is inherited through sex-linked disorder. Okay. So what happens here? Colorblind women, because women normally carry 2 X chromosomes. If you say colorblind is small c, then there should be both defective alleles for a woman to become colorblind, because case here is women carry 2 X chromosomes. To show the problem, they have to have 2 defective alleles. Whereas men normally carry 1 X chromosome, and it is being said that it is normal man. So what do you write? You write capital C with the X chromosome. And always students tend to make this mistake that they probably write capital C or small c for Y, but there is no such gene or mutation possible on Y chromosome. So you don't have to write anything for Y. Only for determining gender, you have to mention Y chromosome here. So colorblind women carries 2 X chromosomes, each with a defective allele. Normal man carries 1 X with normal allele or capital C, and 1 Y chromosome. If you write gametes, both the gametes from here will have X with small c, and here there will be 1 X chromosome with capital C, Y chromosome with none of these alleles. To write children, F1 generation, these two, so there will be capital C with small c, and here there will be capital C, so there will be small c with Y. Small c goes here. So basically, mother is defective. So all males, normally males receive X chromosome from mother, always from mother. Okay, so what happens here? Because mother is defective, mother carries this problem. All male children, whoever receives X chromosome from the mother, they will be carrying color blindness. Whereas in case of females, 1 X chromosome comes from the father. So capital C comes from father, small c comes from mother. They are not affected, but these are considered to be carriers. So they are not affected. If you have to write in the exam, consider them as close to normal. They will not have a problem. They may carry the problem, and these are considered to be carriers. These are known as carriers. So what happens? How many children are affected? If you are asked to write how many children are affected, usually out of these 2 children, this 1 out of 2 got affected. So 50% of children will be affected. If they ask how many girl children got affected, so none of the girl children will be affected. They are all carriers. How many male children got affected? So 100% of them will be affected. All male children, because they have to carry one of the X chromosome from the mother. Because mother is homozygous for this, all boys will be affected. That's how you solve the problem. Okay. So in case if you happen to get a question like that, understand what type of genetic disease they're talking about. Is it autosomal inherited based or sex chromosomal inherited based? And then write about these gametes step by step and the progeny. The biggest challenge will be identifying the genotype. Here clearly they are mentioning colorblind women, normal man, so no other possibilities exist. In case if they ask you to write about, parents who are colorblind have a child, then they are indicating that parents are sending, both the parents are giving those genes. Such hints will be given based on which you have to find out the genotype. Okay, this is about genetics, so know about terminology, mostly terms what we have been seeing in the exam related to this. And Mendel's laws, know about who is father of genetics. Okay, and then diseases, certain diseases that we have talked. That is what I believe are important from this part. Okay, I hope this is useful to you guys. So I'll come back with the other topics related to biology in coming sessions. Thank you.